Hi guys, welcome to the new episode of Archaeal Adventures, where we will be discussing a great new paper on nested symbiosis in halophilic environments. We're going to be deep diving into this great paper published in Nature of Microbiology in October of this year. The paper is from the Krupowicz group from the Institute Pasteur and is about nested symbiosis found in the Danakil Depression in Ethiopia. So in 30 seconds, what is this paper about? Well, in the system there are several layers of symbiosis. Nanohaloarchaea are obligate symbionts of the larger haloarchaea. They cannot perform some essential biosynthetic pathways such as DNA, amino acid, and lipid production. The second layer of symbiosis in this system comes from archaeal viruses that infect either the nanos or the halos, but not both. An important note here is that viruses of archaea are known as archaeal viruses and not archaeophages. These viruses are able to exchange genes with each other due to the cytoplasmic bridges that form between the nanos and the halos. The viruses can therefore rapidly change their capsids and need each other for immune evasion. The final level of symbiosis in this system comes from SCREs, a type of virus satellite found in the haloarchaea. These satellites hijack viral capsid proteins and are unable to spread to new hosts inside of the capsids at the expense of viral DNA. And that was the paper in 30 seconds. You are now more than free to click away. But if you would like to further explore this very interesting system, we're about to dive in right now. In front of you now is the alien environment known as the Danakil Depression in Ethiopia. This part of the African continent is undergoing continental drift, forming a rift valley. This environment is very hot, salty, and acidic, and is dominated by nanohaloarchaea and haloarchaea. The salt content is around 10 times saltier than the sea, with temperatures ranging from 25 to 110 degrees Celsius, and the pH being as low as 1.5. Previous metagenomic work in this region by the same group led to an expansion of what we currently consider to be the nanohalo and the haloarchaea. Currently, there are 3 primary halobacteria genuses and 2 nanohaloarchaea genuses. This study doesn't specify which species are symbionts of which other species, since it is a metagenomic paper. So, what did they do? Researchers took samples at 4 sites across the Danakil Depression in January 2019. Samples were fixed and filtered on site. Some samples were left unfiltered, and back in the lab these were then enriched for virus-like particles before being ultracentrifuged for collection. These collected viral pellets were then imaged using electron microscopy. On the bottom right is figure 1c, which shows some of the viral structures that they found. Filtered samples were also fractioned by size and sequenced to produce a metagenomic database. Viral mags were assembled using VirSorter 2. The CRISPR sequences in the host mags were used to match viral mags to host mags, essentially matching the virus to the host. Okay, so what did they find in their study? Well, the genomes of the viruses reflect that of their hosts, with the GC content being similar between host and virus. Furthermore, the proteomes reflect an increase of the amino acids that are essential for halophilic life. There are several ways in which a microorganism can adapt to a halophilic environment. The species we're talking about today use a salt-in approach, where intracellular concentrations of K+ are increased to balance the proton gradient. This then causes a decrease in intracellular pH. The proteome is then forced to adapt to this by using aspartic acid and glutamic acid more commonly, since they change the protein pI values to reflect that of a more acidic environment. Alanine is also used to combat against the hydrophobic events in such a cellular environment. So what kind of genes do these viruses carry? Well, before anything else, I'm going to mention that the viruses we'll be talking about specifically are the viruses of the nanohaloarchaea, the ones that are symbionts of the larger haloarchaea. These archaea are obligate symbionts because they are unable to biosynthesize amino acids, essential lipids, and nucleotides. Well, naturally, these viruses will carry viral genes essential for their lifestyle, including capsid production, structural tail genes, phage head genes, and replication genes. Nucleotide metabolism genes are also encoded, including RNR, which converts RNA to DNA, DUTPase, which turns DUTP into DUMP. DUMP can then be used to produce DTTP, and thymidine kinase, which is used in recycling T for coming into the cell. For amino acid metabolism, there were only 2 genes found on the viral mags. One was a lysine biosynthesis gene, and the other was an enzyme that turns glutamine into glutamic acid, one of the amino acids needed for halophilic proteins to maintain their structure. And then there were several genes relating to translation. Firstly, there are around 5 different tRNAs that were for aspartic acid and glutamic acid. And then there were genes related to translation efficiency, such as PTH and T4-like RNA ligases. But why do viruses carry these genes? Well, when a virus selects a host, they need that host to be able to produce the viral capsids. By boosting nano-halo-archaeal metabolic potential, they are able to ensure that the capsids get made since the archaea are able to produce the amino acids that they need. Alternatively, increasing metabolism leads to more rapid growth of nanosymbionts infected with the viruses, leading to increased viral spread compared to non-infected cells. Now, back to the haloarchaea and the final level of symbiosis within the system. When a virus infects a cell, it will inject its DNA into the cell, and then that genome will multiply. These genomes will then start to produce proteins which are essential for virion production, such as the capsid and the tail. Viruses then burst out of the host cell and begin to search for a new host to replicate in once more, beginning the cycle again. However, when a virus tries to infect a cell with viral satellites, or SCREs as the paper refers to them, things start to change. The DNA is still injected, replicates, and begins transcription and translation all the same. But when the cell bursts, it is the SCREs and not the viral DNA which is stored within the viral capsids. So further cells are infected with the SCREs and not the archaeal viruses after all. So, in a way, satellite viruses are parasites of the archaeal viruses. They can subvert capsid production and escape from the host to a new halo, but when they infect a cell, they cannot produce more capsid proteins after they replicate. No evidence of symbiosis for the viruses exists, but they have been hypothesized to be advantageous for the halos since they prevent the continued production of virus. Though contrary to this, out of the 9 satellites identified, 7 had anti-CRISPR spacers within the genome of the halos. Moving on to the final finding of this paper, gene sharing between the viruses of the nanos and the halos. When both host and symbiont are exchanging nutrients and metabolites at standard conditions, cytoplasmic channels between the cells open up. These channels are typically around 10 nanometers across. When they are both infected with viruses, the viral DNA is able to pass from one cell and into the other. Viral genomes can then exchange genes. This is figure 5D from the paper. The halovirus EHP23 and the nanovirus DNTV3 share a helping of genes with each other and with other haloviruses. And in figure 5E, we can see that when looking at capsid homology, the nanos cluster at the top and the haloviruses at the bottom. However, we can see some strange things here, such as halovirus HJIV1 and nanovirus NVIV1 clustering together, and 2 more nanoviruses highlighted in red exist within this clade of haloviruses also, suggesting that these genes have been shared from halos to specific nanoviruses. If you're interested in extreme adaptations, I recommend reading the work by Aaron Oren or Purification Lopez Garcia. If, like me, you really enjoy symbiosis, there's a large body of literature that looks at the hyperthermophilic Nanoarchaeum equitans and its host Ignicoccus hospitalis that live in deep sea vents off the coast of Iceland. I hope you all really enjoyed that deep dive into nested symbiosis. Please like and subscribe, follow me on Blue Sky, and thank you so much for watching.